Namaste student, myself J.V. Anshika Kushwaha, Faculty of Agriculture and Veterinary Science, Department of Food and Biotechnology, and our subject is Fundamental of Biotechnology, and today's session is Introduction to Plant Tissue Culture, Cell and Tissue Culture Techniques. So first of all, we will discuss what is plant tissue culture. So plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissue or organs under a sterile condition on a nutrient culture medium of known composition. It is widely used to produce clones of a plant in a method known as micropropagation. Different techniques in plant tissue culture may offer certain disadvantages over traditional methods of propagation, and it includes the production of exact copies of plants that produce particularly good flowers, fruits, or have other desirable traits. To quickly produce mature plants and to production of multiple copies of absence of seed or necessary pollinators to produce seeds, and to regenerate the whole plant from plant cells that have been genetically modified. The next is the production of plant in sterile containers that allows them to be moved with greatly reduced chances of transmitting diseases, pests, and pathogens. Then the production of plants from seeds that otherwise have very low chances of germinating and growing, that is orchids and nepthanes. Then to clear particular plants of viral and other infections and to quickly multiply these plants as clean stocks for horticulture and agriculture. So plant tissue culture relies on the fact that many plant cells have the ability to regenerate whole plant, that is totipotency, and single cell plants cells without cell walls, protoplast. Pieces of leaves, stem, or root can often be used to regenerate a plant on culture medium given required nutrients and plant hormones. So techniques are, what are the techniques of plant tissue culture? Preparation of plant tissue culture for tissue culture is performed under a aseptic condition under HEPA filtered air provided by laminar flow cabinet. Therefore, the tissue is growing in sterile containers such as petri dishes or flask in a growth room with controlled temperature and high light intensity. Living plant material from environment are naturally contained on the surfaces with microorganisms. So their surfaces are sterile with chemical solution. So, what are the applications of plant tissue culture? The commercial production of plants used as potting, landscape, and florist subject, which uses meristem and shoot culture to produce large number of identical individual, and to conserve rare or endangered plant species, and plant breeders may use tissue culture to screen cells rather than plants for advantageous characters. Example, herbicides, resistance and tolerance. Large scale of growth of plant cells in liquid culture in bioreactor for production of valuable compound like plant derivative secondary metabolites and recombinant protein are used as biopharmaceutical, and to cross distantly related species by protoplast fusion and regeneration of the novel hybrids, to rapidly study the molecular basis for physiological, biochemical, and reproductive mechanism of plant, to cross-pollinate distant related and a transformation tissue followed by short-term testing of genetic construction, large-scale production of artificial seed through somatic embryogenesis, and synthetic seed production. So these are the application and introduction of plant tissue culture. Next, what is tissue culture? A tissue culture is the growth of tissues uh, or cells in an artificial medium separate from plant organism. This technique is also called micropropagation. This is typically facilitated by use of liquid, semi-solid, or solid growth medium such as broth or agar. Tissue culture commonly referred to culture of animal cells and tissues with a more specific plant tissue culture and being used for plant. The term tissue culture was coined by American pathologist Montrose Thomas Burroughs. So what are the historical use? So William Rocks removed the selection of medullary plate of an embryonic chicken and maintained in a warm saline solution for several days. So this is the overview of tissue culture. And what is cell culture? Cell culture is a process by which cells are grown under controlled conditions, generally outside their natural environment. After the cell of interest have been isolated from living tissue, they can subsequently be maintained under carefully controlled conditions. These conditions vary for each type cell, but generally consist of a suitable vessels with a substrate or medium that supplies the essential nutrient like amino acid, carbohydrate, vitamin, minerals, growth factors and hormones, and gases like carbon, oxygen, and regulates the physiochemical environment like pH, buffer, osmotic pressure, and temperature. So cell culture now refers to the culturing of cell derived from multicellular eukaryotic, especially animal cells. So this is the overview of animal and cell tissue culture. So what is cell culture? Is a technique where cells grown under a laboratory condition in vitro, and tissue culture is a technique growing tissue by transferring them into artificial environment in which they can continue and function. And organ culture deals with the culture with our isolated organs like roots, anther under laboratory condition in vitro, and different names are given depending on the organ utilized. So this is all the overview of introduction to plant tissue culture and cell culture and tissue culture techniques. Hope you will like this video. And this video is generated by GVW Digital version 2.0. And if you have any query, you can write down in comment section. Thank you.